Thank you so much to Alan and the whole PHA group that they invited me here to this fantastic conference and give me the opportunity to present here. I would like to give you today an introduction into the German perspective of genomic surveillance and focus especially on how research can contribute, or specific public health oriented research can contribute to improve the genomic surveillance. Will there be the slides available here on the monitor? Well, maybe later. So I think it's a little bit late to speak at that conference about what is genomic surveillance, but anyway, this graphic I like quite a lot. So it's an easy thing. So to constantly and systematically monitor pathogens based on their genomic data, and then make these public health relevant bioinformatics analyses to extract exactly that genomic or biology knowledge out of the genomes that is relevant to do that public health related actions. So monitor the disease, control the pathogens, their evolution, give recommendations for interventions and so recommendations to the policy makers, and finally develop countermeasures like vaccines and so on, and end the pandemic or stem out the disease. But some aspects that are not seen in this graphic, which I think from our perspective are quite important to consider. First is funding. So I have just been on the World Health Summit, and there were lots of interesting projects introduced, and lots of them ended with a statement that funding was not enough for either reach the goals at all, or there would be better goals if there would have been more funding, or to create something sustainable. And the genomic surveillance is no exception here. So beside all this infrastructure that we have to implement and find the skilled people that do the jobs we need, I mean, it's based on systematic sequencing of pathogens. And also the sequencing cost has dropped in the last decades a lot. It's still a major factor that makes this kind of surveillance quite costly. So to do a systematic and constantly sequencing of these public health relevant pathogens. And we need that funding not only so from time to time. We cannot say we do a genomic surveillance whenever there is a fund, and if there's no fund, then we stop the surveillance. The next thing is an aspect that we have to consider is the data. So of course the genomic data is the basis of a genomic surveillance. But thinking about genomic data, we have to think about the terms of availability of that data. How do we get the pathogens, and how do we get them sequenced? Where do they get them from? So from a national perspective, it does not make sense to get only from one part of the country lots of pathogens, and from the other not. Then we have, also for the genomics data, we have to think about standards. So you can imagine that just a handful of SNPs in these pathogens that are wrongly classified can make a difference. So in outbreak analysis especially, they have quite an impact. And thinking about different methods of DNA extraction, library preparation, or a sequencing method can produce these handful of SNPs that are probably not, should not be there. The time delay, of course, is important. I mean, outbreak detection cannot wait until we have whatever causes this time delay. But also the conceptual data that make the genomic data so valuable are important to consider. So epidemiological data, the data from the patients, we have to think about include more fields than just the human ones, so from the veterinary side, from the environmental side. But also phenotypic data for the pathogens that are constantly created in the labs has to be as a standard included in the system, as well as clinical data. We heard about data sharing that is essential. So without the willing to share, to perform a timely sharing of the data, the genomic surveillance will not work. And speaking about data management and IT infrastructure, I think the whole genomic surveillance is a big IT project. So we have learned that in our way that we need a lot of skilled people in that field. So kind of data engineers, software architects, software developers. But these are people that are not necessarily related to the life science. So I mean, they get offers from companies like Google and Amazon as well, and you can imagine how they often decide. I mean, money is a big incentive. You will hear this sentence again later. And then public health bioinformatics. It's a special field of bioinformatics that is really concentrating on the needs that we need here. So one thing is that we need in the field of public health bioinformatics standardization. So here is PHA4GE doing a fantastic work that will that will make a big difference and good contribution to all these people that want to establish so national genomic surveillance in their countries. But a topic that I would like to focus today on a little bit more is how public health related or public health bioinformatics related research can improve hopefully a little bit the genomic surveillance. But first I would like to show you our idea of integrated genomic surveillance. The fact of integrated surveillance is often used in another context. So you have heard it in other talks the last days that integrated surveillance means the integration of not only the pathogens from the human side, but also integrating the veterinary and the environmental side. But for us, it is important to see the integrated genomic surveillance as the integration of the genomic data and the epidemiological data of the patient. So that is really essential for us because so alone, both data types are not as valuable as they could be. So SARS-CoV-2 was our pilot project to establish a systematic genomic surveillance. And as I said, we needed genomic data, and we got them from bioprobes that were directly sent to us, and we sequenced them. So from biological surveillance, from laboratory networks that we get support from, or from outbreak investigations and studies where we were involved. But we also got I will go to that into detail in the next slide, genomic sequences from labs from all over Germany, and then we get the epidemiological data or clinical data from the patients from the local health authorities. And that all the data was transferred to the RKI, and there we started our processes to I mean, what you do with genomic data to reconstruct the genomes and extract the biological knowledge and then integrate that with the epidata. So we created that kind of knowledge that we needed to do our task that we did, so which is a surveillance of the SARS-CoV, of the virus, with the detection of mutations, risk assessment of new variants, and the phylogenetic analysis, which were then, of course, the basis for the reporting that we did regularly, and that were also the basis for the decisions often of policymakers. And of course, we also uploaded all the sequences that we got for SARS to public databases. So we uploaded them to 3 instances, to GISAID, to ENA, and we also put it on the GitHub page for easy access for researchers. And without going into detail, one of these instances was not happy that we didn't an exclusive submission to their database. So I told you that we received genome sequences from all over Germany. And you can see that until the end of 2020, in the SARS-CoV-2 pandemic, we were actually quite blind because we got around 100 genomes per week. And that was the time when in the UK they found the first time the alpha variant. So that's no wonder, because the UK started very early to sequence a lot of SARS-CoV-2, and then, of course, they had a chance to detect them first. And then the politicians were very concerned. So they asked us, well, the UK is going on that and that. What's the situation in Germany? And we had to say, we don't know. We don't have any sequences to answer that question. And then that was the time where the situation changed a lot, because the politicians bring into place the corona surveillance regulation, which has 2 implications. So first, by law, all sequences created in Germany for SARS-CoV-2 had to be transferred to the Robert Koch Institute. And second, every sequence that was generated in Germany for SARS-CoV-2 was refunded with an amount of 220 euro. And that was, I mean, that was amazing. So all the people started sequencing like crazy, and there were no complaints at all to about, I mean, data ownership. Everyone was really happy. So it seems to be money is a big incentive. So in total, that changed, I mean, anyway, the situation quite a lot. So from these 100 genomes per week, we increased it quite a lot. In the high times, we received up to 8,000 new sequences per day. And in total, we collected over 1.5 million SARS-CoV-2 sequences from Germany. So that gave us the possibility, of course, to have a very detailed view into the population structure of that during the pandemic, which was, of course, what the politicians wanted from us. Of course, if you think about 8,000 pathogens, sequences per day, then you have to do a lot of automatic workflows to handle all that amount of data. So, I mean, everything, QC, lineage assignments, genomic profiling. We did or established pipelines for outbreak detection, cluster detection, and so on. So creating alignments to produce the phylogenies and finally produce those reports for data sharing. I mean, we developed a lot of infrastructure that, of course, we wanted to use further after the pandemic, and that is not only all the data structures that we created for the data, but also I mean, that amount of data is very essential. You have seen that here, to produce visualization. So very early we started experimenting with next-strain, and there were what is seen there below Heinsberg. That is where the pandemic in Germany started. That was a carnival event. So all the people that were celebrating Carnival after the event were positive, and so that was really easy to see here in the phylogeny. So a dedicated set of samples that is located on one place, and many other events were quite useful. But I don't have to show you the usefulness of genomic surveillance. But anyway, now we experimenting with real-time phylogenies like Usher or Microreact to visualize that going further. One essential point is also to bring the data together and aggregate them and make them explorable. So we found their solution very useful with Tableau, where we could put the data into context and see the associations between the genomic and the epidemiological data especially. As I said, we wanted to use all that infrastructure that we built up for further pathogens. We had already this list, I mean, many years before the pandemic, but we were not heard by the politicians to realize our concept here. But beside the SARS virus, we wanted to have 20 more bacterial viral pathogens from those important categories, so food-borne pathogens, the respiratory pathogens, sexual or via blood transmitted pathogens, and those nosocomial infection pathogens, mostly related with the AMR. So in Germany, we have this law that these pathogens, so whenever there's an infection with one of these pathogens, it has to be reported by law. And you see here the number of reporting cases per year. And for those that are not SARS or influenza, where the numbers are really high, we plan to sequence every pathogen from that case that was reported. So the idea is that the report comes, and then for each of these reports, one isolate of the patient goes to sequencing, and then data together with the epidata will be transferred to the Robert Koch Institute for an integrated genomic surveillance. So that's, in a nutshell, again, what we plan to establish here. As I said, genomic surveillance needs the integration of certain kinds of data, which is the genomic data of public health relevant pathogens. So we get them, or in our plan, we get them from the National Reference Center that have the expertise of these pathogens, but hopefully also from clinics that can contribute, or laboratory networks. Then we need so some kind of good, robust bioinformatic genome analysis extracts exactly those parameters that we need. Well, actually, it's not so much. So it's about similarities, differences, risk profile, virulence profile, IMR profile, things like that, and certain typing schemes that are already very useful if you have that in place. Of course, you can always extend. And we need, as I said, epidemiological data that in Germany we plan to transfer via, there's some kind of digital network called German, I don't know what is the missing. So it's a digital network for the infection protection that is so that encrypts the data and makes all the primary diagnostic labs and all the connected labs available to transmit that data that then can then be transmitted to the Robert Koch Institute. And we will also use that network for the transport of the genomic data. And as an outlook, we would like to further integrate later than environmental data or data from animal and the food sector, and of course data from international IGS systems. We need, of course, some kind of necessary infrastructure. I mean, it makes, as I said, it is a big IT project, so we need a lot of IT structure and pipelines, which does not make sense to create in each of these reference center. So we build up that infrastructure centrally at the Robert Koch Institute, and then we have the ability to decentralize sequencing and make central analysis and dissemination of the data. Yeah, I mean, many IT infrastructures has to be created, data warehouse to integrate all the data. For example, as I told you, these 1.5 million SARS-CoV-2 sequences had to be assigned via, so lineage assignment via pangolin has to be done every night because this classification often changed, and we have experimented with different technical solutions until we found one that is really capable of doing that kind of high throughput action performance. The benefit of a systematic genomic surveillance is quite clear. So, I mean, the surveillance of pathogenic microorganisms and the infectious diseases they cause, and they help us to do our daily work as a National Public Health Institute. We have to detect the outbreak or help on detection of outbreaks and then do outbreak management, transmission surveillance or resistance surveillance, all these things that meanwhile we build an infrastructure that is capable to manage the next pandemic. We use it in these peace times to do the things that I mentioned here. Of course, that amount of data that we collected during SARS-CoV was a perfect database also for applying new models or new data analyzing algorithms from artificial intelligence or modeling that helped us, for example, to predict the number of cases of Omicron several months before. And yeah, of course, countermeasures. And what I would like to say or emphasize is that we are not doing that as an island solution. So we are very much interested to contribute what we achieve on a national level to this super regional surveillance system. So ECDC is planning a system to have a European overview about what's going on. And of course, the WHO with the newly established hub is doing the same. So we would like to build up the national system and then connect it to for getting a wider view on the pathogens. Now let's go to the topic that I told you, how special public health focused research can improve a genomic surveillance. On an example that is Campylobacter. You may probably know, at least in Germany, it's quite an important foodborne pathogen, and it's a zoonotic pathogen with a broad range of hosts, and it's actually in the industrial countries the leading source of foodborne illness. So alone in Germany, we have every year more than 80,000 cases, and that pathogen is often transmitted via the food chain, so undercooked chicken or milk products, and so that's the way of infection. And it was already known before that several lineages, so that based on the 7-gene MLST system, several lineages within Campylobacter jejuni are more or less associated with certain hosts. But of course, the association with these genomic markers are not the reason for why they are associated with that host. And then our question was, could we find a genomic marker that gives us the information about the host that is this particular strain associated with? Where does it come from? So, and for that, we sequenced 500 strains from different hosts. So there was pig, cattle, chicken, and wildlife. So those are ducks. And we sequenced them, and we did genomic analysis that you normally do. So first, we did a pan-genome analysis, which is the core genome analysis on the one hand, and then the accessory genome analysis. So in the core gene of that set, so not the whole population, but our set, from the around 1,700 genes that Campylobacter jejuni comprises, normally we had 1,200 that were available in all of these 500 isolates. So we did a phylogenetic tree, and what you see, if you the nodes of this tree are colored according to the association with the host. So what is interesting is see that for the cattle, these two clades, within the clades they are very similar, so there's not much diversion, but between the clades there's a well, lot of quite a big genomic distance. And the same is for chickens. So we have 3 clusters that are a little bit more diverse within, but they are quite far away from each other. And also we have that pig cluster with the yellow ones in the 12 o'clock. And also a distinct water where these duck samples were located. So that was already confirming the knowledge that there exist different clades associated with certain hosts. And we did next an analysis of the accessory genome. So in our data set, we had around 7,200 accessory genes. So if you imagine then a vector of 7,200 positions, and whenever one gene is present, you put a 1, and if not, then you put a 0. So you can do some kind of principal component analysis to compare these vectors and get a clustering. And we see a similar result here that the cattle strains form distinct clusters. Again, these lila clusters that are quite far away from each other. We have the separated pig cluster, and we can identify also these 3 chicken clusters and the wildlife water cluster below as well. So what we did then is to apply a method which is called a genome-wide association study. It's a method quite widely used in human genomics for cancer research and rare disease research. And actually, it's quite obvious that you should use that also for bacterial pathogens or bacterial genomics. And the idea is to have 2 phenotypes and then have enough genomic data to find the association between a phenotype and changes in the genome. So, for example, if you have the easiest question is in terms of antimicrobial resistance, is that strain sensitive or is it resistant to antimicrobial? And then to compare the genomes, and you probably find the reason, or at least the association between the group of resistant and sensitive antimicrobial or pathogens. So we also applied that method to our need. So we that was the workflow that we used. We used the PySeer implementation, which is quite often used. For our phenotype file of interest here in the example for the cattle strains, the positive phenotype was be associated with cattle, and the negative phenotype was not, was the association with the other host. And then we artificially shredded the genomes into those k-meres in different steps, so from length 9 to length of 100, and made a correction with the distance matrix of the core genome phylogeny to weight the significance of the host-specific k-meres. And finally, we got the k-meres that are associated with all of these hosts. So the next step was then to have a look where in the genome of these strains the found KMAs play a role. So we mapped them back on an artificial genome where all the accessory genes of that data set were present, and then we identified certain regions within genes where these genome mapped, so then they are possible associated elements in the genome that play a role in this host association. So the result from chicken, we found around 6,000 KMAs, while for cattle origin was significantly more, so 60,000 KMAs. And then what we did, we identified those genes where the KMAs mapped, and that was really interesting. Here's an example for the cattle. So you see, and you remember the core genome phylogeny of the whole set. We have these distinct groups for the cattle strains that are very far away from them. But if we take one of these genes that we found, so DNA for DNA replication, and make a phylogeny of these genes, so independent of which cluster they came from, they all have nearly the same sequence. So there is no division into these two clades. So that speaks a lot to be some kind of relevant factor that is associated with host specificity for cattle. And we found another gene, this FFH gene, but just to demonstrate that is really striking the result that gives us a hint that these genes have something to do with it. Similar, we found some targets for chicken and for pig. Also the RPOB gene that we found for chicken to have the same sequence for all chicken isolates. From the E. coli research, it is already known that mutations in RPOB can enhance the growth rate of these strains. And especially if there is a higher temperature than in the humans, so for 42 degrees, for example. And that makes actually a lot of sense if you think that chicken has a higher temperature, so body temperature, than humans. So at least, I mean, there's no proof that this gene is involved, but many findings speak about or speak in that direction, that there could be an association. Also for pig, we found some genes that were involved in the thiamine pathway. That probably has to do with the feeding of pigs that changed a little bit to the diet that they get the rest of the food for the humans. So saying that is that we found targets in the genome of Campylobacter jejuni that can be really good used for a certain purpose. So if you have a foodborne outbreak investigation going on, you first have to link the cases based on the genomic data to see what cases probably belong to an outbreak or to just detect an outbreak. And you, of course, have to do it with the combination with the classical epidemiological data, so questionnaires and that thing, and interviews. But the next thing after you identified a potential outbreak is to manage the outbreak is to find the source. And you can do some kind of intersectoral whole genome data matching if you have available sequences from the pathogen, from the food source and from the patient source, and then you find a match for the similarity. But it would be, often you don't have that situation that you have the pathogens from all these food samples. And if you have certain markers, that gives you a higher probability that these outbreak-causing strain might come from a certain source, and you can more detailed structure your questionnaire and your interviews to ask the patient, have you probably eat undercooked chicken or some milk products, and finding the source much faster. Of course, that is not a definitive value that you can say if that gene is present or that allele of this gene is present, then it must come from there. But it may speed up the search for the source. So it's quite important to take up these genomic markers in the Campylobacter genomic surveillance, for example. So I already come to an end. I mean, you know this slide already from Josefina's talk, but I like this a lot. It's actually very, very useful to think about the strategy that goes on the WHO hub, especially in terms of IPSN. So the Robert Koch Institute is a founding partner of this hub, and not only for that reason we are interested in that they succeed in their activities, but we are also very much convinced that the activities of IPSN are really helpful. I mean, of course, all the nations have to implement the national strategy, but then please try to think on a higher context to follow these meta objectives that makes it easier then to connect the national activities with the national activities from other countries. And then we get hopefully a global view on that pathogen, which makes it so much valuable. I would like to thank many people that I cannot put here all. I mean, for the genomic surveillance, so many people, most of the two biggest departments of the Robert Koch Institute are involved, the infectious disease, microbiology, infectious disease, epidemiology departments. Then for the research projects, we had collaboration with the veterinary faculty from Fire University, and also support from the Federal Institute for Risk Assessment, and thanks to the One Health Approach Campylobacter Research Consortium, and of course for the Federal Ministry of Education and Research for funding the research project, and a special thank to the German Federal Ministry of Health that is financing all the genomic surveillance activities of the Robert Koch Institute. And yeah, I thank you, the audience, for your attention, and that's it.